All righty. So today we're going to be covering chapter 12, adaptive immunity. So this goes with what we talked about last time when we were talking about innate immunity. And we'll be talking about innate immunity again, but now we're moving on to the third line of defense, which is adaptive immunity. And we're going to start with the cells that are involved in this. Okay, so specifically today, we're going to be covering the learning objectives. I'll open it up to questions. Then we'll talk about the cells of the adaptive immune system. We'll talk about maturation of B and T cells. And then we'll talk about activation and expansion of B and T cells. Okay. And then we'll kind of put a pause in the story, so to speak, and talk about the effects of activated B cells and T cells. Okay. And we'll talk about what to do for next time. Okay, first off, this chapter helps us meet the following learning objectives. Being able to define the key role of evolution as it applies to microbiology. Being able to identify microbial structures and connect the structures to their functions. Specifically today, we're going to be talking about, host cell structures, but that still counts. Identify pivotal components of microbial systems important to human health, and analyze and describe the impact of microorganisms. Okay. All right, any questions? Okay, if you think of any, let me know. I'm happy to answer them. Okay, so first off, we're going to talk about the cells of the adaptive immune system. Okay. Now I have antigen presenting cells listed because some of them are a part of the adaptive immune system. Some of them come over from the innate immune system, but they're all important to the adaptive immune system. And we're going to be talking about B cells and T cells. So let us go to the whiteboard. All right. So first off, let's talk about antigen presenting cells and what they are. Okay, antigen presenting cells, also abbreviated APCs. Okay. For the purposes of this class, come in two basic categories. Okay. We have our phagocytes. And not just any phagocytes. Okay, when we're talking antigen presenting cells, most of the time we're talking about macrophages. And dendritic cells. Okay. So these are cells of the innate immune system that their job is to phagocytize. Okay. So when we talk about B cell and T cell maturation, we'll come back to these. Okay. We'll talk about how they present antigens. Okay. And then B cells. B cells are members of the adaptive immune system, but they also present antigen. Okay. So later on, we're going to talk more about what B cells do in their capacity as an antigen presenting cell. Okay. Now, let's go ahead and talk about B cells. Okay, B cells, besides being antigen presenting cells, okay, also, once they are activated, are capable of making antibodies. Okay, antibodies, we've mentioned antibodies before, but let's go ahead and do a little more formal introduction to what antibodies are. And then we'll, next time we meet, we'll be talking about what they do, okay? So chemically, antibodies are kind of globular proteins, okay, when they're just floating around in the blood, which leads to another of their names, immunoglobulins. Okay, oops. Okay, there's no G there. Okay, which gets abbreviated as IG. Now, when we talk about the different classes of antibodies, IG is going to be seen in the abbreviations of those a lot, okay? And it's short for immunoglobulins. Then when they get to a protein, or more specifically, a part of a protein that they recognize. Okay, so let's say I've got a cute little bacterium here, and it has a flagella. Okay, so this flagella is composed of flagellin, but flagellin has all sorts of odd little bumps, okay, that could be seen as different shapes on a chemical level, okay? And let's say that this immunoglobulin recognizes the circular one. Once it binds to that, it unfolds into kind of this funky Y-shaped deal body, okay? So when we draw them in cartoon form, we tend to draw them Y-shaped, okay? And if I were to draw a more accurate depiction of this, we would see something that looks like this. And I talked about this in chapter 2 when we were talking about proteins and quaternary structures, okay, because we've got 3 different types of peptide chains that come together to make the functional protein that is an antibody. Okay, so it's just a lovely example of quaternary structure where you have more than one gene product, okay, coming together to make a functional protein. Okay, so above and beyond that, there are 2 regions of an antibody I want you to know. There's this end here, okay, which is part of the FC portion or the constant fragment, okay. That is recognized, as we talked about last time, okay, last week. This is recognized, this portion of an antibody is recognized by complement. Complement combined to that, and that starts the complement activation cascade. They recognize this constant region, okay? Macrophages, okay, have receptors that will grab a hold of this FC portion, this constant fragment portion, and which enables them to swallow bacteria and other pathogens down better. Okay? Now, on the tips of the Y, this is called the variable region or the FAB. Okay? Okay? When we get into maturation, when we start talking about how B cells learn how to be B cells, we'll talk about how this variable region, for each B cell that matures, we get unique variable regions, okay? So in theory, your body can make antibodies against just about any chemical shape out there, okay? They're very, very specific. Okay, so B cells, their job, once they're activated, is to make antibodies. And we also call this the humoral, okay, response. And humoral refers to blood. Okay, so these guys are circulating within your blood, okay, and other fluids in your body, okay. All right, now T cells come in 2 basic kinds that we're going to be talking about in this class. And those are cytotoxic T cells, Okay, so we abbreviate those as TCs. We also call them CD plus 8s. Okay, this is cell differentiation markers. Okay, we abbreviate that CD, and this is the 8th one that was found. Because when we look at lymphocytes, B cells and T cells, you look at them under the microscope, they all look the same. They all stain the same, unless you use, paradoxically, an antibody stain that will recognize one or the other of these markers. Okay. Now, the other group of T cells that we're going to talk about in this class are helper T cells, okay, or THs or CD plus 4s, okay. And the job of these two, we call this the cellular response or the cell-mediated. Okay, response, because these guys go around looking for cells that are misbehaving. Okay, and then these guys go around and they talk to the antigen presenting phagocytes, dendritic cells. They talk to the B cells. They talk to the CD8 cells. You name it. They are the intermediaries between all of these immune cells that are active in the adaptive immune system. Okay, so all of these guys have something to do with cells. Okay, so that's why we call these the cell-mediated response or the cellular response. Okay, all referring to the same thing. Okay, so these are the major players that we're going to be talking about today. Okay, questions?
Can you go over humoral blood one more time? Was that just the 3 ends of the antibody?
It's the whole antibody.
Okay.
Yeah, the humoral response, when we talk about that, we're talking about antibodies.
Okay.
And because they show up in the blood and because they show up in the body fluids, that's why we refer to it as humoral.
And the THs are helpers?
TH are helpers, yes. T helper cells. Now, when I forgot to give you my abbreviation for antibodies, we abbreviate those as ABs or IGs. And then antigens like the flagella of a bacterium or peptidoglycan or anything that will stimulate an immune response. We refer to that as an antigen. And we abbreviate that as AG. And when we talk about what the antigen presenting cells do, we're going to learn the term. We'll go over the term for these bits and pieces of an antigen. But because repetition is good, these are epitopes. Okay, but we'll talk more about how we get from this big antigen, which is a flagellum, to the bits and pieces, which are the epitopes. Okay, other questions? Okay, let's move on to antigen presenting cell. Well, no, let's talk about how B cells and T cells start to mature. Okay. Let's do that next. All righty. Okay. All blood cells start out in the bone marrow. And there is a stem cell, okay, that is kind of like the founding ancestor of all blood cells, okay, and they are self-renewing so that we can constantly be making new differentiated, okay, blood cells, but we call these hemopoietic stem cells. Actually, I think I got the T and the E in the wrong spot. Let me try that again. Because it's a different poetic. There we go. So we have a bunch of these in the bone marrow. And I really shouldn't even be drawing it red because, you know, not all of them become red blood cells, but some of them do. Through a lot of steps and cell divisions and different signals, they can become red blood cells. Okay? And others go along and they become, you know, eosinophils. Okay? But we're going to be talking about T cells and B cells. Okay, so one of the first steps, okay, is for a cell to decide that it's going to become a leukocyte. Okay. Okay, they've decided that they're going to become this specific kind of white blood cell. Okay. And then the next thing they need to decide is whether they're going to become a B cell or a T cell. Okay, so we're going to say this leukocyte decides that it wants to become a T cell. Okay, and this one has decided that it wants to become a B cell. Okay, now when I say decided, it's not like they, you know, look through a list of things and say, hey, I'm interested in that subject. All about cytokines, cell signals between the different cells at different places and at different times that lead them on this path, okay, of differentiation. But I'm going to use human examples because it's easier to remember that way. Okay. So let's say that the T cells and the B cells have decided that, yes, that is their career path. They want to grow up to be T cells and B cells. Okay. So the B cells stay here in the bone marrow. They stay in the bone marrow to get their education. Okay. T cells leave the bone marrow and they go to the thymus to be educated. Okay, so they've graduated from high school. Now they're going off to college. Okay, so B cells go to the local university. T cells, you know, go across the country because, you know, the bone marrow is a long way from the thymus. Okay, so there's a couple of tests that these guys have to pass for them to be able to go on and graduate, okay, as naive B cells or naive T cells, okay? So in other words, they've matured enough, okay, that they have decided that they are focusing on just one part of an antigen, okay? And this is how they do that. While they are maturing, okay, say B cells in the bone marrow and T cells in the thymus, they do some rearranging of parts of genes in that FAB region, okay? For these guys, it's the FAB region of their T cell receptor. For B cells, it's the FAB or variable region of their B cell receptor, okay? So what they do is they take bits and pieces and they shuffle them around, they snip some out, and they all come up with something different for that variable region, okay, of what will become their antibodies, okay? For right now, it's their B cell receptor. This is showing, okay, this is the kind of antibody that I'm going to make. Okay, so they just, if it's a B cell, they display the kind of antibody they're going to make on the surface of their cells, and we call this a B cell receptor. Okay. And so this one recognizes this kind of shape. Okay, and then we have another one that recognizes, okay, more of a triangle shape. Okay. And the same thing is going on up here with the T cells, okay? But they're not going to make antibodies, okay? But they are going to be looking for specific antigens, just like the B cells are going to be looking for them, okay? But they display what antigen they're looking for, okay, on their T cell receptor, okay? Actually, there's more than one T cell receptor, but we're going to focus on just one. Okay. All right. So these, these are just two examples of T cells that have gone off to college in the thymus, and T cells or B cells that are staying and being matured in the bone marrow. Okay. So there are a couple of questions, like I said, that each T cell, well, let's start with B cells because it's simpler. Okay. They need to be able to say, okay, so the question is, do your antibodies that you're going to make, do they match up with a self-antigen, with a bitten piece of protein, okay, or some other antigen, most antigens are protein, that is made by the body, okay? We call these self-antigens, and the question is, do you recognize self-antigens? Okay. And if the answer is yes, then this cell is killed, it is eliminated. Because we don't want to make antibodies against our own proteins, okay? Because that would mean that we would die. Because we would have our organs, our tissues labeled with an antibody that targets it for destruction by macrophages and cytotoxic T cells. Okay, so that's a bad thing. In fact, it is thought that autoimmune diseases are a failure somehow of this process.
So if it's yes, they kill them?
If it's yes, they kill them. If the answer is no, do you only recognize foreign antigens, then they get to go on. They get to graduate. So for T cells, we've got a little more stringent. Okay, actually, I need to get 3 of these guys up here, because they have 2 questions they have to answer. Okay. And let's go ahead and let's say this one is looking for more of a box. Okay. So the first question that they get asked is, can you recognize MHC1? Okay. In other words, can your T cell receptors shake hands with MHC1? Okay. Now, MHC1 is a molecule, a self-recognition molecule that all cells of the body, excepting red blood cells, make. Okay. And remember, I've drawn it as kind of this M-shaped thing. It's got a groove. And as the cell makes proteins and what have you, they will display bits and pieces of this, okay, in this MHC1, and they'll display it on the surface of the cells. And T cells need to be able to recognize this. Okay? They need to be able to recognize and interact with MHC1 or MHC2. Okay. Now, if the answer is no, okay, let's say this one says, oh, no, I can't shake hands with this. Okay. Then we say, I'm sorry, you're going to have to die. Okay. So we screen for them to be able to do the job, okay, of shaking hands with either MHC1 or MHC2. Okay. Now, the second question that they get asked is the same that B cells get asked. Okay. Okay, you guys, you can recognize MHC1 and MHC2, but are your receptors, once they bind to MHC1 or MHC2, are you going to recognize self-antigens? Okay. Are you going to attack the organs and tissues of this body? Okay. And if the answer is yes, just like the B cells, they are told to die. So once again, when we have autoimmune diseases that relate to your T cells attacking tissues, they shouldn't. It's considered a failure of this process. Somehow or another, something has gone wrong. Okay. Now, this one has answered both questions appropriately, so it's able to go on to become a naive T cell. Okay, it gets to graduate from the thymus. Okay, questions so far?
So what do the B cells have to, so T cells, they do MHC1 and MHC2. What do B cells identify?
B cells, because they're releasing their antibodies out into the environment, they're not necessarily shaking hands. Then they don't have to answer this question about the MHC1 or the MHC2.
Okay. So they just graduate.
Yeah, as long as they don't recognize self-antigens, they get to graduate.
Okay.
Yeah, and they get to go on to become naive B cells. So that means that they have matured, okay? And the T cells, they've matured, okay? And they get to leave the bone marrow, they get to leave the thymus. And for simplicity, this is a simplified version. Okay? We're going to say they all go to the lymph nodes. Okay, that is their next stopping point. Now, just realize the story is more complicated. They could go to other secondary lymphoid tissues, but we're going to stick to just one, just for the simplicity of it. Okay. So before I clear all the drawings, are you ready to go on?
So on the T cells, it's first yes, then no.
First yes, then no yes.
Okay.
And really, I'm pretty sure we know that that's the first test that they have to pass while they're maturing in the thymus. But you don't have to remember it's the first one. Just so long as you know that they have to recognize MHC1 or MHC2 to be able to shake hands with it. And then they have to not recognize self-antigens that are displayed on MHC1 or MHC2.
Hi, ustedes tienen vestidos de...
Okay, you're really quiet. Can you turn your volume up?
Sorry.
Oh, there we go. What was your question again? Okay, if you think of your question, feel free to ask. I'm happy to back up. I just couldn't hear you to start out with. Okay, any other questions before I clear the board? Okay, let's take this up at the lymph nodes. Okay, so I have, and I'm gonna switch colors. Okay, I'm gonna have naive. Okay, CD8s plus 8s. Okay, these are my TC cells. Okay, and then I'm going to do my helper cells in orange. Okay, so these are going to be my CD plus 4s or T helper cells. Okay, and then I'm going to do B cells in this brown color. So these are going to be my B cells. Okay, so they are going to hang out. Okay, waiting for their epitope to come along. Okay, so we're gonna do all of them looking for this circular kind of shape epitope. Okay, now meanwhile, out in another part of the body, I've had a follicular dendrite. Okay, that has, we'll say, for example, we'll get 2 of them going here. Okay, so I've got 2 different follicular dendrites in 2 different parts of the body. Okay, okay, now this one, okay, has engulfed a virally infected cell. Let's say that this is a dendritic cell that is hanging out in the sinuses, and it has swallowed down a cell that has been infected with COVID. Okay, so the respiratory cell will make it a ciliated respiratory cell. Okay, and it got infected with COVID that I'm going to draw as these little round dealy bobbies. Okay, and it was forced to make more COVID, evil COVID. Okay, so it was sick. It was displaying signals that things were not right. And this cell ate it on down. Okay, it was sick, needed to be cleared out. Now, for its MHC1, okay, so this one's going to display MHC1. And I'm separating these guys out for simplicity. But these guys can display and do display both MHC1 and MHC2 at the same time. Okay. Okay, so because this ate it down and it was chopping it up, okay, and it came from the inside. The MHC1 that is displayed, okay, on the surface of this cell is going to be bits and pieces of cell, okay? And it's also going to show bits and pieces of COVID, okay? So everything that comes in here and gets broken down, okay, is going to be displayed on MHC1, anything that comes from the inside of a cell, okay? And this dendritic cell is always also going to display some of its own, whatever it's making, its own proteins, okay? Its own bits and pieces of carbohydrates, okay? Its own little bits and pieces of lipids, okay? So these we call endogenous antigens. Okay. Now, because they get chopped up, okay, in the phagolysosome, that's why we get the bits and pieces that we call epitopes, okay, because you rarely get an entire protein. Now, not all antigens are proteins, but, you know, most of them are. That's why we get bits and pieces, okay? So these are bits and pieces of endogenous antigens, okay? These viral and proteins were made by in the inside of this cell, okay? Now this guy over here, okay, recognizes a bacterium, okay? So it swallows it down, recognizes it as a pathogen, and in the mature phagolysosome, we chop this buddy up, okay? Now, because it came from the outside, we're not talking about self cells, okay, or the proteins made by these cells, okay? So that would include, you know, endogenous includes proteins that are made by our cells, okay? Antigens made by our cells, okay? And that includes viruses, just because of how viruses are made, okay? Now over here, this one is going to be displaying on MHC2, it's going to be displaying exogenous antigens, okay? And they are made by other cells, okay? And so because they came from outside of the body, okay, then they get displayed on MHC2. Okay. So like I said, just as a reminder, this guy on its MHC1 is displaying all the bits and pieces of proteins that it's making, okay? But it's also displaying bits and pieces of this bacterium, okay? So once they have eaten something down, especially the dendrites, okay, come over to the lymph nodes and they say, hi, are you looking for any of these? Okay, so we're going to start out with the T cells, okay? Now these guys can talk to multiple cells at the same time, but just to make it easy, we're going to show this one, okay, displaying its, oh, I got the wrong one. I should have put the image, well, no, I did it right, never mind. Let's get my little guy back up here. So it's going to display its MHC1, and it's going to display the top part of the COVID spike, okay? Now, if this T cell, if its T cell receptor did not recognize this, if it doesn't match up with it, it's going to say, nope, not what I'm looking for. And so this one will go on to the next one and say, hey, you looking for this one? But really what happens is a whole bunch of T cells cluster around these dendritic cells, checking out all of the MHC1s and MHC2s, looking for their antigen, okay? And CD8s look for MHC1 because they're looking for virally infected cells, okay? So these guys, this comes along and shake hands, okay, interacts with this epitope that is displayed by the MHC1 on this antigen presenting cell, okay? But that's not the end of the story, okay? It has another receptor, it's CD8, that has to come and also grab a hold, okay? So that's part of why we call these CD plus cells, is because this receptor comes. And once they both interact, okay, these guys get to talking. They release cytokines and talk to each other, okay, and they actually bind other receptors, but we're not going to go into that, okay? And this cell says, hey, there's my antigen. I need to go out and fight this, okay? I need to fight whatever is making this epitope. But there's just one of me. I need more. There needs to be more of me. And cells in your body can do that. They can divide and make exact copies of each other. We call this proliferation, which is a fancy dancy word for multiplying, Okay. Another term for this is clonal expansion, because we are making exact copies, we're making clones. Okay. So all of these guys have the same T cell receptor. By the way, these receptors are all over their surface, but just for simplicity, I'm only drawing one. Okay. Now, some of them are going to stay back and become memory cells. Okay, they can keep making more copies of themselves, and that's their job. Okay, the rest of them are going to become what we call effector cells. Okay, in the case of CD8, we call these effector cells cytotoxic T lymphocytes. Okay, and next time we meet, we'll talk about what they go out and do. Okay, but it's kind of a dangerous job. They don't last for very long. If I remember correctly, they only last about a week. Okay, sometimes less than that. Okay, it depends upon how dangerous it is and how soon they meet up with the cells that they're supposed to get rid of. Okay. Memory cells keep making more clones of themselves, some of which go off to become CDLs. Okay. How are we doing so far? Okay, if I'm flipping your brains inside out, let me know. Okay. All right, so we're going to get this blue antigen presenting cell. Okay. And it's going to display its MHC2. Okay. And we're going to say that this bit and piece of this bacterium is an exact match for its T cell receptor. Okay, so I'm going to show that interacting with that. Okay, now if I were drawing a truly accurate picture, the COVID round epitope would be a different shape slightly than the bacterial epitope, because antibodies can recognize the difference. They are very, very, very specific. Okay. So once again, not only do we have to have the T cell receptor match the antigen that's displayed on the MHC2, but its CD4, okay, needs to come in and also interact. Okay. If that happens, these guys start talking. Yep. Yep. Okay. And once again, same kind of thing. This cell says, hey, I need to go out and start talking to CD8 cells to get them going, because maybe they haven't gotten the news from this guy yet. Okay, besides, it has to be displayed on MHC1, otherwise they're not going to see it. Okay, I need to go talk to phagocytes to tell them to go out and eat more. Okay, I need to talk to B cells. Okay, so they also make more of themselves, because, you know, that's a whole lot of work for just one little cell. Okay, so I'm drawing three. Really, there are thousands, hundreds of thousands of these clones that are made. Okay, so once again, we form memory cells that continue to make more of this type of cell. Okay, the others become effector cells. Okay, so they can become TH1s, they can become whoops, that's supposed to be a T and not an I, TH2s, they can become T helper repressor cells. Okay, they can become all of these different types of effector cells that all have different jobs. Okay. All right, how are we doing so far? Okay, now I mentioned that B cells can be antigen presenting cells. Okay, say in the lymph node, there's been a bacterium, because we're going to stick to with the bacterium example for this one, that has died. Okay, say because of inflammation. Okay, And there are bits and pieces of bacteria that are floating around. If a B cell binds any of that, it phagocytizes that. Oh, and I'm showing it going into the nucleus because on these lymphocytes, the nucleus is really big. Okay, it gets moved into the cytoplasm, it gets chopped up, okay, bits and pieces, and it will redisplay that bits and piece, okay, on its surface using MHC2, okay? So just for the sake of getting things cleaned up a little bit, I'm going to move this over so I have room to bring my T helper cell over. Okay, we're going to show the B cell receptor. We're up here. Okay, it binds its antigen, moves it in here, moves it back up and displays it here. Okay. So it just kind of goes around to all the T cells, okay? T helper cells saying, hey, do you recognize this? Okay. And the T helper cell says, yes, yes, indeed I do. And they get talking to each other. Okay. And sometimes this cell comes over and does kind of the same thing. Okay. It can talk to these guys even when they're not being antigen presenting cells. Okay. But the effect of this is this B cell gets confirmation that this is really a problem. Okay, because it could be, oh, you know, maybe this is pollen. You know, if I don't ever hear from a T cell, maybe my epitope is a part of pollen and I don't need to worry about it. Okay. Now, some of us, we do respond to pollen, others don't. Okay, so once again, the B cells will do the clonal expansion. Okay. They will make clones of themselves because one B cell cannot make enough antibodies to fight anything. Okay. So they do the same thing. They get memory cells, and the B cell effector cells are called plasma cells. Okay. Now, for the sake of the story, we're going to say that they stay here, okay, in the lymph node, and they just start pumping out the antibodies because they get into the lymphatics and they circulate through the entire body. Okay. They get into the lymphatic tissue, which dumps into the circulatory tissue or the circulatory system. It leaks out of the capillaries into all of the tissues. And so you get these antibodies all over the place. When we test for them, we pull them out of the blood. Okay. All right. So this is the story of maturation, okay, and differentiation and activation of the various cells. And that's how the antigen presenting cells play into this. Yes, Mitchell.
Okay. So the difference between each one is for the T cells that go for MHC1, they only go for MHC1. The helper cells go for MHC2, and then
Yep.
the B cells, they bind to the antigens, and then the T cells or helper cells identify the antigen, and then they get swarmed by white blood cells to eat those.
Yes, well, yes, that's part of what's going on here. This is this guy, this T helper cell is coming over here to confirm to this B cell that yes, you have an antigen. This is your epitope. You need to make more of yourself.
So then it binds to the other antigens, and then
No, just to its own. Well, yes, the antibodies that are released will bind to the other epitopes of the antigen. Yes, once
Okay.
they're released. Did that answer your question?
yes.
Okay, for a minute there, I was feeling like I was talking around your question and not actually answering it.
Maybe. So just to clarify,
Sure.
these don't eat these don't get rid of the antibodies. They just tell the other cells that they need to get rid of, and then the other cells get rid of those phagocytes.
Oh, I tell you what, yeah, you're asking about what antibodies do, okay? And then also what the CD8 cells do, and we're going to talk about that next time.
Okay.
So if I don't cover it, make sure you ask me again. And we'll do a real brief review of this. So how's that for putting your question off?
Good.
So B cells attach to the free-flowing antigens.
Yes.
Okay.
They don't go and talk to the dendritic cells. Just T cells talk to the dendritic cells. So if I were to draw these guys, because remember, all of these are displaying both MHC1 and MHC2. You've got all sorts of T helper cells, CD8s, and CD4s and CD8s coming and checking all of the antigens that are being displayed. And they either say, yep, this is my antigen, and they undergo the clonal expansion. Or they say, nope, this is not my antigen, I'm going to keep looking. Whereas B cells, they're hanging out, and you're right, they are processing soluble antigen that is floating free. Which reminds me, occasionally, B cells do not need a T helper cell to get them going. Okay. And like I said, I've only drawn one B cell receptor. They have them bristling all over themselves like a space satellite. So if their epitope is on an antigen that's kind of big and floppy, and let's say that multiple epitopes bind the B cell receptors at the same time, these guys will be stimulated to undergo limited clonal expansion. And they don't form very many memory cells. So we get a weak antibody response when we have this big floppy antigen that has lots of epitopes that bind multiple B cell receptors. We really get going when T cells help. So even though the B cell is acting as an antigen presenter, it really needs to get that second opinion, so to speak, that confirmation from a T cell, helper T cell, to really get going and making a lot of antibodies. Does that help? I know this is all very complicated, and that's why I try to make a story out of it, because if we have a story that connects to something in your lives that you're familiar with, then it makes more sense. Okay.
Okay, so the B cell attached to those free-flowing antigens that were coming off of the MCH2. Does
No, it would be,
MCH1 have free-flowing ones too?
well, they didn't get detached from these MHCs.
No, they didn't.
Now, this would be like, let's say, off in the tissues, I had a bacterium that complement bound to, and I got a MAC. I get a whole bunch of MACs, and I should have drawn this as a pink cell because MACs are less effective against gram-positive cells, but we're going to go ahead and stick with the color. And this poor guy popped. Okay. And so I now have bits and pieces of peptidoglycan and bacterial DNA and all, flagellin, okay, all sorts of good stuff that comes floating into the lymph nodes and they bind to these B cell receptors.
Okay, so
And-
they bind with cells that have lysed.
Yes, bits and pieces of cells that have lysed.
Okay,
That's one example, but we're going to stick with it. Because it's far less confusing than going through all of the various different permutations of this. But it's bits and pieces of dead cells, yes. Okay, does that help? So this kind of goes with B cells being fluid-based, okay? Whereas these guys being more cellular-based, because these guys, your T cells, cannot see an antigen unless it's presented to them on an MHC. MHC2 for helper T, helper T cells. MHC1 for helper CD8s, cytotoxic T cells. So these guys have to talk to a cell. They can have their antigen come floating past, and it's like they never see it because they can't grab a hold of it. Whereas these guys, I mean, they display what they've eaten on an MHC2 so that the helper T cells can confirm that it really is an antigen, that it really is their epitope. But they grab it out of solution. They grab it out of the fluid. Does that help?
I just like thinking about the cell blowing up.
I know it is fun to think about the cells blowing up. And you know what? It could be neutrophil parts that come floating by, and they won't grab that because anything that would grab a neutrophil part was eliminated in the bone marrow. Because, you know, we don't want to respond to and go attack neutrophils. They're our friends. Same thing up here. If a dendritic cell comes and is clearing out inflammation, and there's a lot of blown up neutrophils, when it displays those bits and pieces, you really don't want a CD8 cell saying, hey, that's my epitope. Let's go kill us some neutrophils. Okay, you're ready to have me ask you questions? All right, let's get some questions going. I have them scattered throughout the PowerPoint because I was going to take breaks, but you know, it's really hard to have a break in the story. Okay, which of the following could be an antigen presenting cell? And I don't know why I have this animated. It made sense to me at the time. Okay, all right, let me get the poll going. And yes, there is more than one right answer. Pick the one you like best. Okay, we've got some good early voting. Okay, we're past 50%. Good job. 75%. Let's get to 100. Hey, we're almost to 100. Keep going. All right, just for the sake of time, let me end the poll and I'll share results. Okay, the favorite answer was B cells. That is correct. B cells do act as antigen presenting cells. Okay, they grab the soluble antigen out, they swallow it on down, present it on their MHC2s, and T cells tell them, oh yes, this is something you should be worried about. Okay, our next favorite answer was dendrites, dendritic cells. Okay, also a correct answer. Okay, they are big time on, sorry, my phone is distracting me. I had it out to keep an eye on the time and the potential spam call flashes on the screen. Anyway, yeah, that's their job, is to swallow stuff down and to display it on both MHC2 and MHC1. Okay, depending upon where it came from. Did it come from the outside? Did it come from the insides of cells that they swallowed down? Or sometimes dendrites get virally infected and they will also display that on their MHC2s. Okay. All right, so we had equal numbers for epithelial cells, macrophages, and red blood cells. Okay, macrophages can be antigen presenting cells. They just don't do it as often as the dendrites do. Okay. They mostly stay in the tissues where they've been and where they've been phagocytizing. Sometimes they leave, but mostly they stay there. Okay, so this is also a correct answer. Epithelial cells, not really. I mean, they do present antigen on their MHC1, but they don't present that to immune cells to stimulate them to become memory cells and effector cells. Okay. So I realize that's kind of confusing, especially when next time we meet, when we talk about how cytotoxic T lymphocytes do their jobs. Okay. But we'll try to make it as clear as anything. Red blood cells, no, they don't even display MHC1 on their surfaces. They are totally dedicated to carrying oxygen, and after about a month they die and your spleen clears them out, and it's a good thing we keep making them from our bone marrow, right? Okay, any questions about why the right answer was the right answer? So just to be clear, the right answer was B cells, dendrites, and macrophages.
Which one of those would be on a test that you'd want us to pick?
So if it was a single choice, multiple choice question, then I would have either B cells, dendrites, or macrophages.
Okay,
I wouldn't have all three. If I had all three, it would be a multiple answer question.
all of the above kind of thing.
Exactly.
Or pick, okay.
Or pick everything that's correct. Yeah.
All right.
Unless I do it by accident, in which case let me know and I will go back and I will make sure you get credit if there's two correct answers on a multiple choice question where there should only be one. I have been known to do that, but I go back and I fix it. Okay, so here's another one. Which of the following adaptive immune responses would be most effective in prevent? I don't want to ask this question yet. Okay, let's skip this one. We'll come back to this one next time. Okay, and I also don't like this one. Okay, oh, and I don't like this question. Yeah, we didn't cover these. Okay, that's cute. Let's see if there's another one. Oh, here's a good one. This one we can answer. And for some reason they are animated again. Don't ask me why. Okay, let me switch. Let's see, let's go up here. Let's get a 4-answer poll going. Okay, so what's the difference between cytotoxic T lymphocytes, those are the effector CD8 cells, and natural killer cells? Okay, now this you're going to have to remember back to last week and go with what I told you about CDLs, cytotoxic T lymphocytes. Yeah, if you're not sure of the answer, that's perfectly okay. I'm going to make you struggle to remember, because when you do get the right answer, you'll remember it longer. Okay, we're at 75%. Okay, oh, there we go, 87%. There we go, 100%. Good job. Okay, the favorite answer was C. Okay, CDL cells use molecules to signal the targeted cell to undergo apoptosis, but NK natural killer cells use phagocytosis. No, this is incorrect. Neither one of them use phagocytosis. Okay. The next favorite answer was B. CDLs target one antigen, whereas natural killer cells are looking for anything unusual. That's the correct answer. Yeah, natural killer cells are looking for either a reduction in MHC1, okay, on the cells that they're checking, or they're looking for MHC1 that is displaying something that they recognize as a microbe-associated molecular pattern. And so and then CDL cells are looking for just their epitope. They're scanning the MHC1 for just their epitope, okay? They don't care if they come across a cell that doesn't have an MHC1, they say, not my job. Natural killer cells can take care of that, okay? But if they come across a cell that's displaying their antigen, their epitope, they're part of an antigen, then they will tell the cell to die, okay? The next favorite one is natural killer cells are granulocytes and T cells are lymphocytes. Okay, not quite. Okay, they're both lymphocytes, but natural killer cells are a part of the innate immune system, and an awful lot of the cells of the innate immune system that we talked about were granulocytes. Okay, so good job on that one. Nobody picked A, and that's also a correct answer. Natural killer cells are part of the adaptive immune system, or excuse me, cytotoxic T lymphocytes are a part of the adaptive immune system. Natural killer cells are a part of the innate immune system. All right, any questions about why the right answers were the right answers? Hey, well, like I said, we'll come back to those questions that apparently I decided not to cover those topics after I wrote those questions. But here's what to do for next time. As a reminder, the embedded tutoring assignment number 3, the last embedded tutor assignment for the semester, is due on Friday. So that means if you want to meet with Rochelle real time using her Collaborate session links, you have Thursday and Friday to be able to do that. Okay. If you are wanting to make use of the discussion board, okay, you really need to have submitted your question on Friday. If you haven't done that, and you do want to use the discussion board, go ahead and submit it today. Some points are better than no points. And you never know, Rochelle may be able to have enough interaction with you that you can get full points, and inevitably you'll also get your questions answered. Okay. All right, and then if you haven't done so, read chapter 12. I realize there's a lot of brand new information, okay? But it's all very interesting brand new information. So hopefully, it'll be interesting reading, and repetition really does help, okay? All right, we're getting done a little bit early, not too bad, but any questions?
Are we going to go over the information so we can answer those questions, or?
Yes, next time we will
Okay.
go over the information that you can answer those questions, and I'll move those questions over to the PowerPoint for next time.
Okay.
Yeah.